Hello everyone. Today I'll be presenting my project titled Drug Interaction Prediction using Graph Neural Networks. So, drug-drug interactions occur when two medicines react with each other in a way that can reduce effectiveness or cause harmful side effects. This is especially risky for patients who have multiple medications such as elderly patients or people with chronic illness. Since testing every possible drug combination clinically is expensive and time consuming, my project focuses on predicting potential drug interactions computationally using deep learning and molecular structural analysis. So, the main problem is that the traditional DDI detection methods are slow, manual, and very incomplete. Existing computational models either rely heavily on external biological data or fail to model how two drugs interact as a pair. So, my goal of the project is to predict whether two drugs may interact, use only their molecular structure, provide a fast, scalable, and reliable prediction system. At the core of it, what we are using is the Siamese graph neural network. Each drug is represented as a smile notation. I will be showing the smile notation now. This is how a smile notation looks. That given data set we have chosen in the CSV file has about 3.5 lakh test data set, train data set I mean. So each of them are labeled as 1 or 0 based on interacting or non-interacting. This represents one of the smile drugs, and then this other thing is the other notation. So the smile drugs are essentially simplified molecular input line entry system. That is what they're called. So each drug is represented using a smile notation, which is then converted into a molecular graph. We are using the default libraries like RDKit. So what it essentially does is extracts the smile functions and then converts them into atoms and molecules. Atoms are nodes and bonds are edges. Instead of treating drugs independently, our model processes two drugs simultaneously using a shared neural network. This is the Siamese variation. This allows the system to directly compare their chemical structures and learn interaction specific patterns. So, how this works is the input smile strings are converted into molecular graphs using RDKit. Atom level features like atomic number and bond type are extracted in this form and then made into a graph. So, this then is processed into the Siamese GNN encoder. So, how that works is essentially both drugs are passed through the same GNN encoder, ensuring consistent feature training. The learned embeddings of both drugs are concatenated to capture pairwise relationships. A neural network essentially then predicts the interaction based on the probability of interaction between the two drugs. This architecture improves the generalization and avoids bias by using separate encoders. We are doing match-wise. This is the model architecture we have been using. We have run it for about 30 epochs. Now let us just show the graphical representation and the GUI approach of this. So this is our GUI, so drug interaction predictor. Autofilling for basic two molecules to show demonstration of how the output might look. It analyzes it. We are using the backend model .pth file. As you can see, it shows us that these two molecules, which are ethanol and methanamine, have low interacting probability, and it also gives you a probability score. It uses an API called PubChem API, which essentially like extracts how the molecule looks. It also gives you a description and context about what the molecule exactly is. So our model is successfully able to predict whether two drugs are able to interact or not. Uh, Let us take one more example. This is one of the other smile string. I'm just going to copy and paste. Similarly, I will copy the other smile string here and paste it as the molecule B, analyzing the interaction. This should again provide us the probability and how they interact along with the diagrams. As you can see, on the right side, the system displays the predicted interaction probability. In this case, it shows a low interaction probability of 30% score. Below this, there are the molecular structures for each of them, along with their IUPAC names, which improves interpretability and user understanding. So the model was evaluated as such, using like the standard metrics like accuracy, precision, recall, F1 score, etc. We got about 83.7% accuracy, F1 score of 0.84, and ROC AUC of 0.91. These results show that the model performs well even on unseen drug combinations, which is crucial for real world use because traditional systems fail in this case. Uh, this project can be especially useful for doctors and pharmacists during prescription, maybe like not completely medically but more for academic research purposes, healthcare systems for safety checks, pharma companies during drug development, as well as risk assessment. It helps in early detection of harmful drug combinations, improving patient safety. To conclude, this project demonstrates how graph neural networks and Siamese learning can be effectively used to predict drug-drug interactions using molecular structure alone. The system is able to scale, interpretable, and suitable for real-world deployment, making it very valuable to the healthcare AI. Thank you so much.